On this episode of Shareable Science Beyond the Blog, let's take a look at the first results from human trials for a vaccine for COVID-19. Welcome to Shareable Science. Science you can share. Within the last few days, we've received some of our first peek at the progress towards a vaccine with at least a couple of the promising candidates. We've got two of them that we're going to talk about today. The first is from the U.S. biotech company Moderna, and they released their interim phase 1 results, so a piece of the data. And this story was shared through press release and an investor call, so we don't have a paper with lots of data and tables that we can dig into. But here's what we know. Number one, the vaccine protected lab mice against lung infection, so it works in animal trials. Number two, when the vaccine was given to human volunteers, it did generate an immune response. That's the goal of what a vaccine does. It primes the immune system to be ready if it encounters the virus. And this is the piece that we know. There were 45 volunteers in this study, and all 45 of the volunteers were found after they'd been vaccinated to make antibodies that recognized and bound to the SARS-CoV-2 virus, so binding antibodies. A subset of those 45 were further studied to actually look and see if any of those antibodies actually are neutralizing antibodies. And this is the key. It's nice if you have antibodies that recognize and bind the virus, but what you really want are antibodies that recognize and bind and prevent the virus from getting into a cell. They block its entry. And of the 8 individuals that we have data on, they made these neutralizing antibodies that in a lab test blocked entry from the virus. Now, we don't know anything about the 37 volunteers that aren't described as making neutralizing antibodies. It doesn't mean they didn't, they just weren't described in the results. So, promising early data from Moderna. The University of Oxford, in partnership with pharmaceutical company AstraZeneca, also released data. They released preclinical data looking at the impact of their vaccine on preventing monkeys from developing the virus. So they vaccinate the monkeys, and then they wait an appropriate period of time, a couple of weeks, maybe a little bit more, and then they intentionally expose them to the virus. And the data showed that for these monkeys, the vaccine prevented them from developing lung infection, from developing pneumonia. So the serious complications associated with COVID-19, this candidate seemed to do its job. The challenge is that when they looked in the nostrils of these monkeys, there was still a high amount of virus. In fact, the same amount of virus or a comparable amount of virus as what you would find in monkeys that hadn't been vaccinated. So it doesn't completely prevent the virus from taking hold, and these monkeys potentially could pass it on. Now, that doesn't mean you're going to find the same problem in humans. It's an issue potentially of timing and of dosage, but it is something that we've got to pay attention to. Okay, so those are the early results from at least 2 of the key candidates. Now, let's talk about what happens when we get to the final phase of a vaccine trial, when we have to look at testing thousands or tens of thousands of individuals. Traditionally, we take all these volunteers and they're either given the vaccine or the placebo, and it's blinded to them. They don't know which one they get. And then they're allowed to go about their daily business for months or maybe even for years. And the hope is that some of those individuals will come in contact with the virus and the vaccine will give them protection. So at the end of this window of time, you actually say how many people who got the vaccine and who got the placebo ultimately got infected, and then you do your analysis. The problem is that in a pandemic, that window of time is too long, and we need to think about ways to shorten that up. So there are 2 potential ways that we could explore trying to streamline this process. The first is emergency use, and so you would give the vaccine to individuals who are already, because of their daily activities, at a higher risk of developing COVID-19. For example, healthcare workers who come in contact with infected individuals on a regular basis. So you would use them to determine if the vaccine was beneficial. The second is more challenging, and it's actually called human challenge. And this is when you take young, healthy volunteers and you vaccinate them, and then you intentionally infect them with the virus that you are studying. And the benefit of that is you get that information in a much sooner time frame. But as you can imagine, there are all sorts of concerns about how you would manage that. How would you recruit? Do they know the risks that they would encounter? And this has been done before in other vaccines, but always when there was an effective drug to treat the individuals that actually got the infection. We don't have that set of drugs right now with COVID-19, which makes this even more challenging. So stay tuned. We'll see how these ultimately play out. But that gives you the background on early data on vaccines and how we think about trying to shorten the time frame. I hope you find this useful. If you do, consider sharing this with those around you. Thanks for watching. I'll see you next time. Stay safe.